Hello, AP Bio. Today we are going to talk about a signal transduction pathway specifically related to COVID-19. So let's go ahead and get started. In this lesson, there are a set of guided notes that you can use to take notes and to answer questions that will help you to understand this pathway, and those will be linked below. So to get us started, in order to understand this particular mechanism of infection for COVID-19, we have to talk about the immune system. So we're going to talk about the immune system first to then talk about how COVID-19 bypasses your immune system and affects your body. So there are two basic types of immunity. You have the innate immune system, which is your generalized immune response to infection. This includes things like your skin, your mucous membranes, and different secretions. And it's considered your sort of rapid response team for any kind of infection. But then you have your adaptive immune response. And this adaptive immune response is a recognition of something you've already been infected with before. And this can include antibodies that can defend against infections, can be cytotoxic cells that can also defend against infections. So there's a variety of different pieces to each of these types of immunity. We're not going to go into the entire immune system today because that's its own video. And luckily, there is also a video that you can watch that will help you with this. So there's a Kurzgesagt video that you had in one of your video quizzes. And if you are not in my class, then I encourage you to go watch this Kurzgesagt video on the immune system, because it's going to be really, really helpful for understanding how the immune system works in relationship to COVID-19. So please go back and review that video if you haven't already seen it. But before we get into that, I want to ask you this question. Most mammals have a twofold response to infection, and that evolved in most mammals. So my question is, why do you think that this evolved? What do you think is the evolutionary benefit to having a twofold immune response? So if you said anything related to the idea that you can be more efficient at defending against an infection if you already know what the infection is, then that's a great way to respond to this question, because your skin and certain immune cells, they can only do so much. But if you already have a library of infections that you have seen, your body's going to be much more prepared for you to defend against those infections. This is how vaccines work, which we're going to talk about how vaccines work in this lesson today, right? Think about it as if your body has its own library of information that it can draw from when it's trying to defend against certain invading pathogens. Speaking of pathogens, let's talk about a little bit of terminology that we're going to need to be familiar with in order to understand this lesson. A pathogen is anything that is going to invade the immune system, and that's a virus or bacteria or any kind of invading microbe. An antibody is a molecule produced by an immune cell in response to a pathogen, and we're going to go into this in more detail later on. An antigen is the molecule on the surface of a pathogen that your immune system recognizes as an invader. Usually this is a lipid or a carbohydrate. So if we look at the following examples, we have measles, chickenpox, the flu. COVID is another example of a coronavirus. The flu is another coronavirus or another new pathogen. And we have vaccines for all of these viruses. Measles has a different antigen compared to chickenpox, and our bodies produce different antibodies in response to those antigens. But if you are getting infected by a new pathogen that your body hasn't seen before, then you don't immediately have access to that antibody. And that's going to be a little bit of what we talk about in today's lesson. So one more resource that you can use to help you remember the players in the immune system is this Bioman Immune System game. There is a link here to the activity worksheet that you can use to help you practice understand the immune system, but it's not required. It's just if you feel like you need more practice with the immune system. Okay, so let's take a look at an example of innate immunity and an example of an infection. So I want you to take a look at this diagram and describe what each of the cells appears to be doing in this diagram. I'll give you a second and jot down a couple of responses. So this is an example of a pathogen that is invading because of a splinter. Now, in our environment, there are all kinds of invading pathogens that could potentially invade our immune systems. But the only way they can get into our bodies is either via piercing of the skin, so you bypass that innate immune system barrier in the skin, or through our bodily orifices, so through our mouth and through our nose, sometimes even through our eyes. But in this case, we're talking about a microbe that invaded by bypassing the skin layer. And there are a couple of different cells we're looking at. We're looking at a mast cell, we're looking at a macrophage cell, and we're looking at a neutrophil cell. And it looks like, based on this diagram, the mast cell and the macrophage cell are secreting some sort of substance that is involved in getting rid of this pathogen. And if you notice, the macrophage actually consumed one of the invading pathogens. And it looks like these signal molecules, they aren't even touching the invading pathogen. These signaling molecules are going into the bloodstream. So this indicates that the mast cell is producing signaling molecules that recruit something to help get rid of the infection. Maybe it's recruiting this neutrophil. So let's dive into these types of cells for a second. Neutrophils are going to be these suicide warriors in the bloodstream that kill pathogens. So neutrophils are cells that hang out in the bloodstream ready to kill pathogens, and they will consume pathogens and then commit quote unquote cell suicide in order to get rid of those pathogens. And in the process, they actually release a bunch more signaling molecules that will recruit more of your adaptive immune response. A macrophage is sort of the first responder. It's the EMT of your bloodstream. It's going to be the first thing that recruits the neutrophils to the infection if the infection becomes worse. And these macrophages, if you go back to the Kurzgesagt video and review the Kurzgesagt video, they actually can wrap themselves around the invading pathogen and eat it via endocytosis. So as we go through these processes, it's going to be helpful to keep track of which cells are part of which process. So which cells are going to be a part of innate immunity, which cells are going to be a part of adaptive immunity, and which cells could potentially be a part of both. So there's a table for this in your lecture guide that you can use to answer this question, or you can write this down in your notebook. Another type of cell that comes from the Kurzgesagt video is a dendritic cell. So this is the intelligence officers of your immune system. And they're found in the bloodstream, and they collect data by destroying the pathogen and then presenting the antigen on the surface of this cell. And they travel throughout the immune system after they have received this antigen, and they present this antigen, and they start looking for the right helper T cell. So these guys, they are the background researchers. They are in the bloodstream. They find the appropriate pathogen, and then they say, okay, we've got this information. Now we're going to take this information and give it to the helper T cell. Now, the helper T cell is the all-purpose commander cell, and it's going to activate the other types of your immune system cells. This is the B cells and then the macrophages to recruit additional macrophages. These helper T cells eventually can evolve or adapt into memory T cells, which guard the tissue against that same invading bacteria or pathogen. B cells are going to be the excuse me, I just had to fix one of these slides for a second. B cells are going to be a type of white blood cell that makes infection-fighting proteins called antibodies. These antibodies can then be used to recruit more of an immune response, or they can be used to create memory cells. So there's two different types of B cells. Plasma cells are going to be antibody factories that are specific to an infection. And so they're going to be the ones that recruit an increased immune response to the site of infection. Memory cells are going to hold on to the recipe of that particular antibody in the case of a future infection. So this is an innate immune response, and we have an example here of a white blood cell chasing after a pathogen, in this case it's a bacteria, and swallowing that bacteria in an attempt to get rid of the pathogenic infection, right? So could you categorize all of these different types of cells? Try to go back and look at the different cell types and consider if they are part of the innate immune system or the adaptive immune system. Did you get it right? Take a look at this chart and consider how you categorized the cells and how this chart categorizes the cells and how it might have been different compared to what you did. So, how does COVID-19 get past all of these immune protections? How does it get past these different responses to an infection? Well, if you've learned anything about COVID-19 in the past, you know that COVID-19 has the following structure. It's basically a sphere of membrane, of phospholipid membrane, that has proteins and carbohydrates sticking out off of the top. And on the inside, it has RNA. We call these proteins, we have different names for the proteins sticking up off of the top of COVID-19. These are the E, S, and M proteins. Now, each of these proteins does something slightly different for COVID-19, okay? And I encourage you to draw the structure in your notebook because this structure is going to be important for later drawings and understandings, okay? The one protein that we are particularly interested in is the S protein. And these are these red proteins here that kind of jut off in the viral corona. The reason it's called a coronavirus is because it looks like it's got a little halo. And the S proteins are what give it that halo, right? That sort of protein layer around it. Now, COVID-19 is a type of RNA virus, but it's not the first RNA virus that we are familiar with. RNA viruses have been around for a long time. And part of why they are so infectious and common is because they are so simple. They are literally just a membrane, proteins, and RNA. Some examples of this are the flu virus. So if you've gotten a flu vaccine every year, you are defending yourself against a rapidly evolving flu virus, influenza virus. And HIV is also an RNA virus, and they have very similar structures that are slightly different from COVID-19, but that basic phospholipid layer, protein, and RNA is all the same with all these different viruses. Now, one of the debates about the COVID-19 vaccine was how quickly it was produced. But the problem is the COVID vaccine was in development for a very long time. RNA vaccines had been in development for a very long time. Operation Warp Speed was the acceleration of those research projects. And basically, they just got a bunch of funding to do the experiments that had already been being done. And so nothing was accelerated unsafely. It was just they were able to go through the necessary steps in a faster manner because they got more funding because of the COVID-19 pandemic. Now, one of the interesting things about RNA viruses is that they evolve very quickly. And the way that these viruses evolve is they evolve from mutations. So because these viruses are not alive, they have no mechanism for defending themselves against mutations, whereas most living organisms have the ability to adjust and fix mutations. But in a virus, they don't have the ability to do that. So the environment, such as radiation or toxins or vaccines, they can cause the virus to manipulate and mutate very quickly. And this is where so many of the variants have come from. So, what is it about the viral structure that makes it likely to mutate? Compare it to a eukaryotic cell and think about, why is eukaryotic genetic information not as susceptible to damage? Think about this structure for a second while you pause the video. How did you answer this question? Well, if you said anything about the nucleus protecting the DNA in eukaryotic organisms, you would be right. One of the reasons that viral RNA mutates so quickly is that it doesn't have any kind of additional protections. It's just protected by the membrane, right? RNA is also single-stranded, which means that it is less stable and more susceptible to mutagens being things that cause mutations. Its simplicity, its short sequence, also means that it is much faster at reproduction and making more viruses. So this rapid rate of reproduction also causes it to mutate much quicker. So here's another question for you, just to check in and consider where we are at in this lesson. Which part of a generalized invading pathogen will be recognized by a macrophage? And what is the structure called on a COVID-19 virus? Now, you might have to go back and review the immune system for this, but take a second to answer this question, and we'll see you in the next video.